Anther. Anther is the male reproductive floral whorl. To know about the structure of flower, click on the link above. Androecium is made up of definite number of individual members, called stamens. Many stamens combine to form the androecium. A stamen consists of mainly a filament, and an anther. The anther is usually dithecous, which means it consists of two lobes. Internally, this dithecous anther is tetralocular, that is, it consists of 4 chambers, called microsporangia, or pollen sacs, or pollen chambers. The process of formation of pollen grains is known as microsporogenesis. Microsporogenesis takes place inside the microsporangia, or the pollen sac. Ts of an anther. Parts of an anther are epidermis, endothecium, middle layers, tapetum, microsporangium, and pollen grains. Epidermis. It is the outermost protective layer. The cells of epidermis are flat and compactly arranged. Endothecium. It is a single layer which is present below the epidermis. These cells can absorb water to help in dehiscence of anther when it matures. Middle layers. These are present below the endothecium, and are formed of 1 to 3 layers of parenchymatous cells. The cells of these layers degenerates at maturity. Tapetum. This is the innermost layer, and provides nourishment to the developing microspores. The cells of this layer may be multinucleated. It is also involved in formation of spore pollenine, microsporangium. An immature anther contains massive diploid sporogenous tissue. These tissue undergo mitosis, or they directly function as MMC, that is, microspore mother cell. At maturity, MMC undergo meiosis to form 4 haploid microspores. These are generally in the form of tetrahedral tetrads. Formation of microspores by meiosis of diploid MMC is called microsporogenesis. Structure of pollen grain. Pollen sac, or the microsporangium, consists of male gametophyte, called pollen grains. Each pollen grain is unicellular, uninucleate, spherical, or oval haploid structure. The wall of pollen grain has 2 layers, called sporoderm. The outer layer of sporoderm is known as exine. Exine is thick, and highly resistant. It may be spiny, or smooth. Exine is composed of sporopollenine, which provides resistance to pollen grains, from physical and biological decomposition. Exine has depressions in 1 or more places, which look like small pores, called as germ pore. In monocot plants, pollens are uniporate, that is, they have only 1 germ pore, while in dicots the pollens are triporate, that is, they have 3 germ pores. The inner layer of sporoderm is known as intine. Intine is composed of cellulose and pectin. Development of male gametophyte. Microspore, or pollen grain, is the initial cell of a male gametophyte. The development of male gametophyte is endosporic, that means, it occurs within the microspore. It involves 2 mitotic divisions, both at different places, that are, before pollination in the pollen sac, and after pollination on the stigma. Before pollination. Inside the pollen sac, the pollen grains divide by mitosis, to form 2 unequal cells. The smaller cell is called as the generative cell. The generative cell has a larger nucleus, thin cytoplasm, and it lacks reserve food, and vacuole. It also lacks definite cell wall, and is freely suspended in cytoplasm of vegetative cell. While the larger cell is known as vegetative cell, or tube cell. This cell has large vacuole, cytoplasm, nucleus, and reserve food. In most of the angiosperms, pollens are released in this stage, that is the 2-celled stage, after dehiscence of anther. The 2-celled stage pollen grain is also known as young, or partially developed male gametophyte. After pollination. The two-celled pollen grain gets deposited on stigma. On stigma, it comes in contact with the sugary secretions, and absorbs it. Due to this, the volume of cytoplasm increases, and creates a pressure which acts on inting. Thus the inting of pollen grain comes out of the germ pore in the form of a tube called pollen tube. The tube nucleus, cytoplasm and generative cell, all migrate into the pollen tube. The pollen tube grows down, towards ovule, through the style due to chemical stimulus inside the ovary. During this the generative cell divides through mitosis, in the pollen tube to form two haploid, non-motile male gametes. This represents the male gametophyte. This completes the male gametophyte in its development. The next part will include structure of an anatropous ovule, and its development.